get this started. So today we have Anna Klompen. Is that right? Did I say that correctly?
Yes.
Cool. She is currently a Chancellor's Doctoral Fellow at the University of Kansas in Ecology and Evolutionary Biology. After receiving her Bachelor's of Science in Chemistry and Biology at the College of William and Mary, when she is not caring for the different jellyfish species maintained in the lab, she is exploring what jellyfish venoms are composed of and how jellyfish use their venom for specific ecological interactions using bioinformatic, phylogenetic, and functional genomic approaches. So really excited. I've been looking forward to this talk all year. Without further ado, Anna, I think you can start.
Great. Well, thank you very much for having me too. So I'm going to try and hide some of these things here. Oops, I hid the wrong thing first. So give me 1 second. I'm usually on 2 screens, so this is a little bit different for me as well. High floating. Okay. So this should all begin there. So hello, everyone. As introduced, kindly, my name is Anna. I'm a PhD student at the University of Kansas, and yes, University of Kansas, pretty much middle of the US, where I study jellyfish venoms in a primarily jellyfish lab. So including actually some of the ones that you're seeing here. So these are just some moon jellies that we had in the lab at one point. Moon jellies have that little cloverleaf pattern in the middle, not too long of tentacles, but those 4 cute little oral arms. So those 4 kind of frilly bits in the center. So we actually maintain and keep 7 or 8 different species of jellyfish in the lab. And that's how we do a lot of our experiments. And for me, because mine is, I'm looking at essentially their venom, so how they sting. Mostly I'm going to be talking about just one of the species that we care for, but I've thrown in also another project I worked on a different jelly that some of you might be a little bit more familiar with in the area. But with that, I'll just give a general introduction to jellies and their stingers and what venom is, and then I'll talk about my 3 different projects, but I'll try and do some pauses here in between for any questions that anyone has. So this is a jelly that I'm guessing many of you are familiar with. So while I'm doing my PhD here in Kansas, I'm originally from Ohio and actually spent many, many summers at Hilton Head. I actually have family that lives in that area. So I'm somewhat familiar, I think, with where you all are at and in Hilton Head, and these were definitely a common sight over the summers. So some people call them cannonballs or cabbage head jellies. I know you get huge bunches of these over the summer, and they're often described as non-stinging. They can sting, so we'll get into this in a moment. All jellyfish can sting, absolutely can sting. They just all can't sting humans, and this is an example of these. But when I say jellyfish, a lot of people think of something like this. The reality is that everything you're seeing on the screen in some way could be called a jellyfish. And I'll get a little bit into why jellyfish sometimes is actually gets a little bit more complicated for some people, but I'll explain how I use the word use the phrase jellyfish and what I mean by jellyfish. But you can see there's all these different crazy shapes, colors we have up in the corner. So something that looks like an egg, that is called an egg yolk jellyfish. It actually eats only other jellyfish and it gives it that yellow color. Down at the bottom part, we have coral reefs and anemones. So corals and sea anemones are in the same group as jellyfish. And over in that bottom left-hand corner, we have something that looks like a long stingy noodle. So that's a deep sea siphonophore, or this deep sea large jelly. Now I think recorded as one of the longest animals on the planet. I believe there was one of that actual species recorded over 150 feet long. But you can get jellies that really are as small, if not smaller than your pinky nail, and only live for maybe a few days to a few weeks, up to jellies that might live. Immortal jellies is maybe something you've heard of too, but jellies often, you can get them living up to 5 years. So the average is probably around 1 or 2. But anyway, all these wonderful things that are considered jellies. But I want to kind of put into kind of categorize these in some ways, because I think that's going to be helpful for just understanding where my work, what my focus really is, and also what a jellyfish is. So this is called a phylogeny or a tree diagram. So all these diagrams show is kind of the relationships between species, the relationships between things. So for instance, in the middle, in the yellow and red scyphozoans and cubozoans, you'll see have only this one connection between them. So they're pretty closely related. But if you think about, say, the scyphozoan and cubozoans all the way to what's called anthozoa, so that bottom one there, you have to go really, really far back into this tree to actually find a connection between them. And so this helps us kind of determine who's most closely related to who by essentially how close the connections are. Now, these are all the fun scientific names of the phylum Cnidaria. So this is the group in which jellyfish are in, but I'm going to go ahead and change to probably names maybe you're more familiar with, and actually I'd use more commonly. So hydroids and hydromedusae, we'll hear a bit more about these, but these are also called the water animals. These are very clear and still stingy animals that you'll see some pictures of in a moment. Stalked jellies, which will also show you they're super cute little things that hang out in kelp. True jellies, and then box jellyfish, which you may have heard before. And on the bottom here, you'll see coral sea anemones, zoanthids, all of those kind of belong in their own little group. And then there is this funky little set of parasitic jellies that I won't talk about here, but I'm happy to answer questions about if you have them. So it could be when you say jellyfish, you kind of mean this whole group of things, this whole groups of cnidarians, or but when I'm thinking about jellyfish, I'm mostly honing in on these four colored groups at the top, these hydroid, stalk jellies, true jellies, and box jellies. They fall into this group called Medusazoa. Maybe you've heard of Medusa before. So Medusa is another way of saying jellyfish. So these are all groups that at some point have had a jellyfish in their lifetime, jellyfish, as opposed to corals and sea anemones, which never really have gotten a jellyfish. And so that might be when you refer to jellies, maybe you're just talking about this little group here. And just to show you some of those pictures that I had before. So all these at the top in the blue boxes, these fall into this hydroids and hydromedusae. A lot of them are very clear, a lot of them really funky looking. The more traditional jellies you might think, or at least the more hardy ones I think about, are these true jellies. So moon jellies also fall into these, as well as those cannonballs. These things called blubbers, that egg yolk jelly, and then sea nettles, which some of you may be familiar with, fortunately or unfortunately. And then box jellyfish. So these are these really dangerous stingers that are typically in these warm water areas. So these, this one here is the Australian box jellyfish. We'll talk about this one in a little bit later, but renowned as one of the most dangerous animals on the planet for humans, at least. And then this cute little tiny box jelly that's actually from Hawaii and kind of more in those areas, actually in the Hawaiian, that tropical areas there. So you could say that jellies really are just kind of in this group here. You could say jellies, all of this. It all gets kind of confusing again. But when I say jellyfish, I'm really referring to a very specific stage of the animal's life. So in fact, kind of like caterpillars and butterflies, jellyfish is really more the equivalent of the butterfly in that system. And actually, they spend most of their life looking completely different, actually looking like something called a polyp. So a polyp, so this is for moon jellyfish, their polyps kind of look like little sea anemones. Or almost little plants. They actually stick to the bottom, so they're stuck to either rocks or sediments or maybe the deck. And then they stick their tentacles out. They have a little mouth here, and they can spend most of their life essentially as a polyp. But then when conditions are either very good or very bad, moon jellyfish make this little pancake stack of different jellies. And each one of those kind of individual pancakes is a very, very young jellyfish. It actually has a whole different name because it looks nothing like these jellyfish at the top. But jellyfish, and when I say jellyfish through this talk, I'm talking about this really specific stage where you have something that is swimming around, has these tentacles, and this bulb here. This is a jellyfish. And it is part of the life cycle that many of these species go through. Okay, I think I might pause there for just a second before I start getting into toxin or venom stuff. Is there any questions right here while I take a drink?
No, I think you're good to go.
All right, awesome. So I showed you a whole bunch of weird and crazy looking things, and beautiful as well. How are they all connected? So all of these animals, these things again called cnidarians, are also called the nettle-bearing animals. And nettle refers to these kinds of cells that they have called stinging cells. So this is what enables jellyfish to actually sting things, is they have hundreds, if not thousands, if not tens of thousands of these tiny little cells that they can use to catch prey or deter predators, called stinging cells. So I guess you call them scientific name, their formal name are nematocysts, are these kind of structures in these cells. You might have also heard the term cnidae before. So nematocysts are a category of cnidae. And cnidae is what all cnidarians have. That's what puts them into that group if you have these stinging cell structures called cnidae. But this is just a fun picture of one that I took from a jellyfish that is not supposed to be very harmful for humans, and it doesn't catch anything that's super fast moving. But you can just, I hope you can appreciate just how complex this structure is. So this is the capsule where all of this material was kind of condensed together beforehand, but when it fires, it has this portion here with all these barbs and spines. So this is the part of the stinging cell that actually kind of would get embedded into the predator or prey that you would have. And these spines would help hold it there as this thread. So this huge long, I think it just goes even farther out of focus into the camera. That's what goes into the actual prey item, so the other animal, and the venom will come out of the end, wherever that end may be. And this is just one kind of stinging cell. Some stinging cells look quite terrifying close up. So this is one of the largest and most menacing, I would say, looking of stinging cells. They have a huge kind of harpoon-like structure with lots and lots of these spines and barbs, and then a long thread that, again, if I was to take this thread all the way out, it would probably go off the screen for a good portion. These are only found in a few different species, and they are found in ones that need to catch really fast moving stuff. So you can imagine if you're an animal that can't move super well, or you're super delicate, you would want something really strong like these to be able to attach onto prey. And these are just a few other pictures of some from actually my animals that we'll talk about in a moment. So all these little capsules right here, these are a bunch of different stinging cells. And this is just one portion of the animal. All right, so this stinging cells are the ways that cnidarians, or this group that includes jellyfish and corals, actually use their venoms. So, but what is venom? And I'm guessing a lot of you have heard about venom, or at least venom and poisons and whatnot. I think I've heard that that's something that's been talked about before, because venom is found in many, many different animals. But what is it exactly? It's just this complex mixture of toxins. So this mixture of chemicals that that cause some sort of harm, that are importantly injected through some sort of really special mechanism, be it fangs, a stinger, or stinging cells. And for that animal with venom, it's serving some sort of ecological purpose. So it's often for predation, it could be for defense. There's a few other ways that some animals use their venom too. But that is what venom is. And venom itself has actually evolved well over 100 times in all animals. So this incredible kind of chemical weaponry has appeared many, many times over the animal tree. So this is a much larger, more complex tree. I don't expect you to read all of these names or all these scientific names on here. All I really wanted to show here is that where all these colors are, these kind of respond to those ecological purposes, these roles that venom may play for each of these groups. And you hopefully can see, and unfortunately, I think there's only a little green here. So hopefully this is easy enough to see. Vast majority of venom use is for predation. So actually for the animal to feed on some sort of other animal. Defense, so protecting the animal in some way, is also fairly common in here, but not nearly as common as the predation. And this is really nice to look at if you're interested in kind of overall trends. So if venom has appeared so many times, is it actually being used in the same ways multiple times? But it actually gets a little bit more complicated. So there's actually different venomous animals. So some of these as an example that use different venoms. They produce different toxins for specific purposes. So for instance, this is a cone snail here. So this is the end of the cone snail here. And this is an assassin bug. So both cone snails and assassin bugs actually make a predation venom and a defensive venom. And they can decide, they essentially flip between which one that they want to use depending on the situation. So for instance, if these cone snails are actively going after fish, they're going to sting it and inject a certain kind of toxin. But if, say, you accidentally have a person that accidentally picks them up because they think they're a beautiful shell, they go into a defensive mode and use an entirely different set of toxins that are actually much, much worse for people than if they had stung them with those toxins meant for them to catch prey. So this is a really important, but also kind of, if you think about it, a crazy thing they're doing. So they're not only making all of this complex toxins for some purpose, they're actually making two different kinds, and they know when to use each. Assassin bugs are the same. They have two different kinds, one for predation and defense. Scorpions also somewhat do this. They actually have a pre-venom, so a sting that has a different set of toxins that they might use more as a defensive maneuver, and then a full sting, quote unquote, using their full venom repertoire would be perhaps for predation. And then the other thing that might happen is actually a venomous animal might change its venoms as it ages. So we know this happens actually in many different spider species and snakes. So you might have heard that younger snakes have a different set of toxins than when they're older. And that's true for some species. It's also true for spiders. So they actually adjust the kinds of toxins they make depending on their age, because you can imagine a really young spider or a young snake is going to be eating something different or have to defend itself against a different set of predators than when they are adults. So this kind of trend has been really, is kind of a hot new topic in venom research. And there have been a few people that have studied this in cnidarians, but mainly just in sea anemones. So this is 3 different kinds of sea anemones here. And there's been some work looking at essentially not necessarily if they produce, how they produce different kind of venom repertoires, but just essentially looking at which toxins they put in which parts of their bodies. Because remember, they're in these little stinging cells and they can move those cells to different parts of their body. And so that means they have some control of which stinging cells they're going to put where. And so in some cases like these, so stinging cells they put in their tentacles are different than the stinging cells they're going to put in the mouth, in the central part here. And in fact, when you look at the toxins just in the tentacles versus the mouth, they are different. And even with this sea anemone here, so this is a Nematostella, either also called a starlet sea anemone or the digging sea anemone. They actually, similar to spiders and snakes, will change their venoms as they grow up, kind of in response to either eating different things or different predators that they might encounter. But I just told you I was telling you a lot about jellyfish, right? I wasn't really talking a lot about sea anemones. The fact is that there's very, very little study in looking at jellyfish. So all these different kinds of crazy jellyfish and all the different ecological roles they might play, and all these different like predator and prey interactions they might have, there's not a lot of work on what their venoms look like. So this is the same kind of tree that I showed in the beginning, color-coded the same way, just with those scientific names from before, but just box jellyfish, true jellyfish, stalked jellyfish, and these hydrozoans here. All this group of medusa or jellyfish-bearing groups, there's very, very little work done. And that's essentially where my research comes in. So I'm trying to apply the same things that have been studied in sea anemones, looking at how their toxins might be different used differently and applying that to jellyfish. And perhaps you're asking, well, that's a good scientific argument, but why me? Why am I particularly studying jellyfish? So I have really loved jellyfish like since I was quite young. And I was just fascinated by these like beautiful kind of alien-like creatures that you would see. And the fact that they had, in most cases, people are afraid of these, right? A lot of people are afraid to go in the water because of jellyfish, amongst some other things, but jellyfish is a common one I hear. And I always thought it was so amazing that there's these animals out here that have such like interesting ways of interacting with the world in terms of stinging things. And I thought, of course, these must be well studied. We must know most of what's happening in a lot of these animals, especially ones that sting people pretty frequently. And it turns out we really don't. There's just not a lot of people studying this right now because jellyfish can be a little bit difficult to work with. But I just found them so fascinating early on. And this is actually, so this is a picture, I think 3 or 4 years ago on my birthday, I was actually trying to move and get pictures of this lion's mane jelly. So some of you might know lion's manes. These are very big animals and can be very bad stingers. I ended up stinging my wrist, actually, just trying to get some pictures of this one. But I found that was a perfectly good birthday present for me. So I was very happy with the situation. And just some of these maybe I thought very basic questions were, so what are jellyfish venoms made of and how do they use them? So what of all these different kinds of toxins that are out there and we know that are used by many different venomous animals all across the animal groups, what do jellyfish have and what are they using them for? Mostly to eat, capture food, or to defend themselves. Why are some jellyfish species so toxic, especially to people? Because to my knowledge, no jellyfish naturally is a predator of humans, and yet we get these really strong responses to some of these species, but not to others that are really closely related. So why is it that some are so toxic? And then this last one here, are there actually different strategies for the ways that they sting things? So we think of making contact with a tentacle, and that being the primary way that at least humans get stung, or that they want to sting prey or predators and whatnot. But are there different ways that they do it? And in fact, there's a very different way that one species, at least, and probably a couple others, use their stinging cells in some way. All right, so this is just outlining some of those what I thought, again, were like basic questions. And this is the 3 different projects that I'm going to talk about today. But I think I'll pause again in case there's anything. Take one more sip of water.
I have just a quick question going back to cannonball jellies. Why is it that they are unable to sting us?
Yeah, no, that's a good question. So that whole group of jellies that have those really like frilly bits and not many tentacles. So they're all that frilly stuff is actually a whole bunch of mouths. So the general group for those jellies are like the many mouthed jellies. So they are filter feeders primarily. So they're eating very small things to filter through those many very small mouths. So they do not have very large stinging cells, one, for most part, because they don't need to catch anything that big. And the other thing is that because they have all this extra tissue around, I think they just don't, the stinging cells are not as condensed. They're more spread out in many ways. So you're not getting a lot of contact on the skin. One, and they're just not big enough. Your skin is actually pretty thick, and most jellyfish can't sting you just for that purpose, including most of the jellies I work with. I can handle them with my hands just fine, and they're stinging me almost certainly, but they can't get through your skin. So that's what I suspect is true with the cannonball jellies.
Cool, thank you.
Mm-hmm. All right. I'll keep on rambling on about these. So these are, of these 3 topics, this first one here is going to look at pretty closely the animals I work with pretty much every day. And it's going to be asking both like what are the toxins that are in this animal, but how does this really cool feature that this animal has called a division of labor, how we can use that to study what these venoms actually do. So these are the jellies that I really commonly work with. So they're called Hydractinia symbiolongicarpus, and that's a super long name for what actually is a pretty small animal. So these are polyps. So this species actually has lost its ability to make a jellyfish. It no longer does that. It just stays as a polyp. And each of these like little individuals here, so these like little flowery things here, those are only probably about 1 millimeter in size to give you a sense of scale. All that you're seeing here, so all what looks like individuals, this is actually one colony of interconnected polyps. So while it looks like there's maybe 40 or so different animals here, this is all technically one animal. They just have each of these polyps that kind of act independently. And so we can keep these animals pretty happy in the lab. And the reason I was super excited to work with these animals is because they have this thing that I mentioned earlier called a division of labor. So you see these polyps with the, I'm actually not sure that you're able to see my cursor. Are you able to see my cursor? I apologize. I was doing a lot of movements with it before.
Yes, we can see your cursor.
Awesome, great. So you have these, they almost look like Hydra, if you're familiar with the freshwater hydroids, Hydra. But then you have these weird other, almost, I think of them like Christmas tree looking ones here with these really speckly tops. So this is an entirely different polyp type. So there are multiple polyp types in this one colony. So again, one animal, but they do very different, have very different functions. So this one here, the one that looks a bit like Hydra with the long tentacles, these are feeding polyps. We call them also gastrozoids, but these are primarily the feeding animals. So they have these tentacles to catch prey, and they have a mouth right in the very center. These other Christmas tree looking ones, though, they do not have a mouth, and they do not participate in prey capture. They actually actively avoid prey. When I try and give them food, they will shrink away from it. So these are called gonozoids or reproductive polyps. So again, they don't feed, they don't catch prey. Their sole purpose is just for reproduction. So you might have males or you might have a female colony, and that is all that they do. And then not shown here, there's actually a third type that also does not eat, does not eat, it does not have a mouth, it actually only participates in prey capture. So all it does is actually, it's these long spiral guys called dactylozoids, or we call them also spiral polyps. They actually will fling their whole bodies almost like a slinky, like a string, catch food, and then pass it on to these feeding polyps here. So again, these are all the same animal, but they do drastically different things on that animal. So for me, when I was coming into the lab, what was really exciting is that, oh, I could just look at what toxins there are in these 3 different polyp types. So similar like the sea anemones I told you before, they move all cnidarians, all these nettle-bearing animals move their stinging cells to different tissues that might do very different things. So like the tentacles versus the mouth, for example. But in this case, we have these entirely different polyp types. So they are probably moving specific sets of stinging cells that have their own kinds of toxins for predation and digestion. So they use them for digesting as well. Defense, since these polyps don't have a mouth, and don't participate in prey capture, I assume that they would use their toxins for defense. And then for these dactylozoids, just prey capture. So toxins that are just important for catching prey, but not necessarily for digesting it and feeding like it is for the others. So this was part of the work that I was going to do. So I was lucky coming into my lab is that we already had a little bit of data to play with. So Dr. Steve Sanders was a PhD student in my lab 5-6 years ago. And he was just interested in general, what are the differences between these three polyp types? So we already had this set of data looking at the genes that were expressed. So genes being these really important key pieces of DNA that are expressed that actually might translate into a thing. He was looking at which genes there were expressed between each of these three different polyp types and what were the differences between them and if that made some sense. So essentially, I just went in and looked at the genes between these and I asked, so which of those are toxins? And can we see if the toxins specifically are different between these? And in fact, we could. So this is going to maintain the same kind of color coding for these three different polyp types, where this purple is the feeding polyps. So these ones that eat. The red are these just predation only polyps, and then the blue is the reproductive polyps. Not much blue though, as you notice. There's mostly, for all these different kinds of genes, you're going to see a lot of purple and red. So what this graph is showing, so each of these kind of names is just essentially the function or the way that these toxins work. So you have allergen related toxins, so toxins that really target your immune system. You have toxins, membrane active or pore forming toxins, so toxins that essentially punch holes into cells. And you might have neurotoxins, so toxins that attack your nervous system specifically. So there's a bunch of different categories here. And then on this axis is just the number of genes. So it goes all the way up to 10 here and just 5 here. So not too many genes, but still quite a significant number that are in these feeding polyps. So again, these are each of these genes, I should say, means that some toxin in this group is highly expressed or exclusively in this polyp type. So the feeding polyps have 2 of these allergen toxins that are only found in this particular polyp type, not in these, even in the same animal. So the same animal doesn't have it in these kinds of polyps. Many different auxiliary things. So these are toxins that just help all these other toxins function. A lot of membrane active or pore forming toxins and a lot of neurotoxins. So a lot of these things that we know are important for catching prey in other venomous species. But the exciting thing for me was that there's several of these predation polyp toxins that are specific to this particular polyp, that are in this pore forming neurotoxins, and then this thing called hemostatic hemorrhage. These are things that attack your blood, essentially, that are really specific to predation polyps. And this was very exciting because that means, yes, this animal is kind of dividing its toxins between how they're used in the environment. Predation for predation only is very different from if you need to both catch food and digest it. And there's not very much that these other kinds of polyps, these reproductive polyps, have. And that's not too surprising. Again, they're not participating in prey capture. They're just acting defensively. So maybe they're just using the few toxins that they have. They don't need that many to be able to protect themselves. So all of this was very cool. This is the first time this has been shown in any real medusozoan. So again, that whole group include all these different animals had never been really looked at before to see if animals change their toxins in some way. But there's a pretty easy explanation for what might be going on. And so we wanted to kind of rule that out before going farther in this. And it could be that it's not actually that these different polyp types have different toxins, but that different stinging cells have different toxins. So I showed you a bunch of pictures of stinging cells earlier, but there's actually well over 30 different types of stinging cells, and each cnidarian species has a specific set of types of stinging cells that they might carry. It's actually a way that we can identify species is based on the kinds of stinging cells that they have. Usually they have anywhere between 2 and 4 different types in their body. So one explanation could be, well, these different polyp types don't actually have different toxins. They're not moving them in some different way. They just have different stinging cell types that they carry. So it could be that these guys have one type of stinging cell, these have another type of stinging cell, and these have another type of stinging cell. That's almost the case. So these are just graphs that show kind of the proportion of each of 3 different types of stinging cell. So I have some kind of graphics here of what we're talking about. So this gray box, these so-called desmonemes, these actually don't have venom at all. These are essentially Velcro. They're super, super sticky stinging cells that make it really hard for animals to move away, and they just kind of hold prey still. These things called uroteles, we'll just call it these full stinging cells. These have those barbs, those spines, and they inject venom just like I showed from the beginning. So these feeding polyps, actually, oh, and I should say too, I apologize. So as you kind of go down the line for each of these bar graphs, essentially I'm just moving down each area of the polyp. So the top parts of each of these is talking about what stinging cells are in the tentacles. For this one here, hypostome is just a fancy name for the mouth. Since this is the only one with a mouth, it's the only one of these 3 that has it. Mid body column, so just the center portion of each of these, what stinging cells are in the center. And then the proximal just means lower. So what stinging cells are in the lower portion of each polyp. And again, this is from 0 to 100% for each of these different types. And what we found was that the feeding polyps have a ton of these sticky desmonemes, these sticky ones that don't or should not have venoms, at least not that we know of. And this is a kind of a close-up of the tentacles here, and all these little dots that you see, those are the sticky, sticky stinging cells. This is another close-up of kind of these here. Only when you get to the mouth do you start seeing these stinging cells that actually would inject venom. So this kind of circular round one called the urotele again, but they kind of look like a crescent moon shape. So we got this bizarre situation where the feeding polyps don't have that many stinging cells that actually inject toxins, supposedly. They just have a few around the mouth and kind of across the body. But it gets weirder when you look at, remember, those reproductive polyps versus the predation polyps. This was the ones that was so exciting because the predation polyps seem to have a bunch of toxins that were really key for catching food. But in fact, they have the exact same stinging cell type. So the tentacle, these top parts here for both these polyp types are exclusively these large uroteles. So just to give you a sense, these scale bars here are the same in all 3 pictures. So these super tiny stinging cells here that are in the feeding polyp are just these massive, massive round capsules in both these other types of polyps. So we do not have, essentially different stinging cells with different toxins is not a good explanation because it doesn't explain why these predation polyps had some toxins that were really important. And actually that's super cool. So that means that these animals are not only kind of deciding which stinging cells to put in different places on the animal, but they're actually kind of setting aside a certain stinging cells with certain toxins. So getting even more in the nitty gritty and then making sure that they go to the right polyp types to do a specific function. This is a really complex thing that they're doing. So I just want to emphasize that too. A lot of people think jellies, this whole group, they are pretty simple animals, but they're doing really, really complicated things. They're just different than many other animals are doing it. Okay, so this is I revealed it twice now, but the next part is pretty exciting. So now we had a bit of a problem. So I had all this data, again, from a previous student that had all this different gene expression stuff, so looking at different genes of the different polyp types. But my stinging cell data didn't really correspond very well. So ideally, what we would want to do is just look at the genes in stinging cells, or at least all the cells that were going to become stinging cells. This would help remove a lot of the noise, but it also really help us hone into getting more specific about what really are true toxins in this group. So to do that, we did something really, really cool that we could only really do with a few different species, including Hydractinia, so my hydroids here. And that's we made a transgenic animal, or a reporter line. That means I literally took a gene for red fluorescence, so a red glowing gene, and I inserted it into my animals and essentially said, okay, wherever there is a stinging cell, I want you to glow red. and just in the stinging cells. So these are pictures of the results of those animals. So everywhere you see one of these little circular red pieces here, that is a stinging cell that's either been made or is being made right now in the animal. They actually make their stinging cells in that center portion, and then they move up into the tentacles. So this had never been done either, looking for stinging cells on these animals. So this is already crazy enough. I came into this lab like not knowing that this was going to be possible. But then we did something even crazier. So there's some super fancy machines out there for looking at cell biology, including these machines that you can essentially ask it to look at. You can ask it, can you take everything that glows red and pull it from everything else that's not red? Essentially, at least in my animals, can you pull out all the stinging cells that I've now made glow red and leave everything else behind? And that's exactly what we did. So now we had a really, really pure sample of just stinging cells from my animals that then we could look for genes, look for these toxin, these venom genes for, which was very, very exciting. So this is just another set of pictures. So this is with a little bit of a fancier microscope. So again, wherever you see red, that is a stinging cell, kind of either made or developing, growing. Wherever you see blue, that's actually just indicating a cell is there. So this is a good check for us. So this is actually the edge of the animal. So this is almost kind of, it's, excuse me, not skin, but it's an extra layer on the animal protecting it. Lots of blue and no red. That's a good sign. That means that this is marking cells that are really there, but we know the red is just marking my stinging cells, which is all this stuff in the middle here. They make tons of stinging cells all the time. And you can watch this in real time, pretty much. It's super, super cool. So this is that center portion of the animal. These are the tentacles. And even in this, you can actually see these stinging cells here poised and ready to be fired. It's super, super cool. So I have a ton of fun just watching these animals, let alone the research I get to do with them. So you don't need to memorize all the names. So these are all these different groups of toxins that I found. I found a little over 100. That's about what we would expect for jellyfish. So that was a good standing text. So their venom, at least for these stinging cells I'm looking at, a bit over 100 different toxins making up this venom. What I just want to show you, so some of those groups again, so again, all these allergens, so this is a proportional chart too. So several kinds of allergens, lots of these kind of helping toxins do what toxins need to do. A few of these pore forming toxins, so we're going to talk about those in a little bit, but not very many. And then a good number of neurotoxins. So all of these different types of things that we see in other venomous animals. So we're feeling pretty good about that. But then the next thing I did is I did the same kind of study that I showed in the beginning with a bar graph, where I essentially asked which of these toxins is specific to each of these polyp types. Remember, again, the whole point of this is to try and figure out which toxins are used for specific ecological roles. And that would be between each of these different polyp types. So I only did the comparison between the feeding polyps and the reproductive polyps. I didn't include those predatory polyps, and I'll talk about why in a minute. And essentially what I found is that comparing these two, all of the top, like 51, this is a slightly reduced number just for some logistical reasons, but well over half of the toxins were specifically in the feeding polyp as opposed to these reproductive polyps. So again, I think that just shows like kind of what we saw before. The reproductive polyps don't need that many specific toxins. They can just go with a few that these animals, either the feeding polyps, the predatory polyps, are already making, and just use it for defense. They don't need to use much else. Whereas feeding, active feeding, and digestion, these are really important functions. And so you need a good suite of toxins to help you out with it. All right, so why didn't I include the predatory polyps in there? In some ways, that was the most interesting one. So this is where the biology of this animal kind of throws us for a loop. So Hydractinia is also called snail fur hydroids. because they grow on the shells of hermit crabs. So these are Pagurus-longicarpus hermit crabs. They are found all really through the northeast coast of the US, and anytime you find them, you will find hydroids on their back, and it's going to be pretty much my hydroids. So not only is that super cool, but the slightly annoying part is that dactylozoids, so those predatory polyps that you see kind of dancing over here, they only grow at the opening of the hermit crab shell. We cannot grow them in the lab, and you cannot grow them without the hermit crab in the shell. So we have a really, really hard time getting enough of these polyps to actually do research with them. We're kind of working on that right now. I just want to show the hermit crabs are super cute. We have hermit crabs in the lab. We've been trying a bunch of different things to try and grow these kinds of polyps, to do some work on them. But that's still kind of ongoing. But it is a fun kind of problem to solve, because we get to work with this whole different kind of interaction that most people, I think, don't have to worry about with their other animals. We have to add another one into the mix. Okay, I'm going to take a drink of water. And we'll get going to some really toxic animals. So I'm going to go a little bit quicker through these, hopefully, because I know we're running a little low, or I've been rambling on. So why are box jellyfish so toxic? So I talked a little bit about box jellyfish in the beginning, and especially this one, the Australian box jellyfish. But box jellyfish as a group, so cubozoans, so there's roughly about 60 different species. Almost all of them have super, super potent venoms, at least towards people. These tend to be the big problem species in many cases, and these are the animals that can send people to the hospital pretty routinely if you get a really bad sting. And in particular, this Australian box jellyfish, so the bell of this animal, this part can be as big as a volleyball. These tentacles, I believe, can be about 10 feet, and there's tons of them. I think it's just 2 meters, so about 6 feet of tentacle contact with you is enough to kill an adult in under 3 minutes from heart failure. That is incredible. And again, this is why this animal has been labeled one of the most dangerous animals on the planet for humans, just in terms of the venom potency. Things like snakes and a lot of other species cause a lot more harm more routinely, because we're not encountering these kinds of jellyfish all over the planet, right? These are really restricted into Australia. Box jellyfish are everywhere, but this one is the really dominant one in terms of medical relevance. But the reason that these toxins are so bad seems to be this really specific group of toxins called jellyfish toxins. And yes, that's an extremely non-descriptive name in some ways, because aren't all toxins jellyfish have jellyfish toxins. This is just the name that researchers have been sticking with, because we haven't done a lot of work on this group overall. And so I'll talk about that in a moment too. But jellyfish toxins is this group of toxins. They were initially isolated from different kinds of box jellyfish that are highly, highly hemolytic. So they attack blood cells really, really specifically, and heart cells really specifically. So it was thought that these toxins, just them being in box jellyfish, is why they're such a problem. But over the years, there's actually been many other kinds of species, including those that are not harmful for humans, that seem to have these jellyfish toxins in them. So then that kind of raised a question immediately to me, was just how common are jellyfish toxins? We now have a ton of great data on a bunch of different species, but no one had systematically gone to look just for how common these things were, and really to test if box jellyfish were really alone in having these. And then we want to know how they were related. So let's say there are jellyfish toxins in a bunch of the species. Are the box jellyfish ones special in any way compared to all these others? And then from there, can we determine why it is that box jellyfish are so dangerous? So I'm going to spoiler alert the 2 questions right away and essentially tell you jellyfish toxins are everywhere. So here's another one of those trees, and you are not going to be able to, I don't even want to read these bottom portions of these. I looked at over 150 data sets from these, and from the roughly 20 to 30 jellyfish toxins that we knew about, mostly from box jellyfish, I found over 111 new jellyfish toxins, all across different species, including moon jellyfish I showed you before, including hydra, that freshwater jelly I talked about before, tons and tons of species that definitely, some of them may be bad stingers, but they're not nearly as bad as box jellyfish. This tree diagram is essentially asking that second question. So how are they related to each other? If they're everywhere, what is something weird just happening with box jellyfish? So again, I have all these, the scientific names again for all these cnidarian groups, cubozoans in red being the box jellyfish. So also a red on here. This yellow, it should say scyphozoans, is this yellow one here. Those are true jellyfish. So your cannonballs, moon jellyfish, sea nettles, all that good stuff. And then this blue are the hydroids. So what my Hydractinia are in, as well as hydra, siphonophores, those kinds of weird things. And really just what I want you to see from this is that we got a massive group of red right here, a little group of red right here, and then these kind of blue and yellow scattered in between. So essentially all of those dangerous, pore-forming jellyfish toxins from box jellyfish grouped together. That's pretty cool. That means in some ways they're all related to each other more in some ways than to any of these other kind of random jellyfish toxins there. That's a good sign also medically. That means that this group is what you need to worry about in terms of medical stuff. And actually everything in this top portion here, we call this JFT1. So this group has the again, naming is not the sweet for, for some of us scientists. So just JFT1. Everything in this lower group here, completely new. We don't know if these toxins are super potent. We don't even really know what these toxins do. So I'm not going to talk about these in the bottom, really. I'm just going to talk about this group that has this massive bit of red at the top. So if we zoom in on here. So because we had all of this red, all of this box jellyfish that all do these crazy potent toxins at the top, we were able to do a few other kinds of tests since we can see a pretty strong relationship. They all group together. And essentially we found out that all of these toxins in this dangerous group are evolving faster than any of the toxins in any other part of these trees. So any of these other toxins in these other species. Usually when we see kind of this increased rate of evolution, so this faster rate of evolution, that usually means that there's some sort of ecological reasoning for it. So, or it's benefiting the animals in some way to have this increased rate of evolution. Box jellyfish prey on fish, things with backbones, so vertebrates. Whereas many of these other species, some of them do, but definitely not to the degree that box jellyfish do. So right now we think maybe all of these dangerous toxins really are because box jellyfish move towards eating fish. Since fish are kind of more closely related to us and many other things, their toxins just accidentally do really medically terrible things for us, as they might do for fish. So kind of an interesting way to think about it, just that their toxins have really adapted to this predation, this type of prey that they want, that has this accidental effect on us. But we never really noticed that it was just kind of this accidental thing that box jellyfish did, because we never looked for toxins in all these other groups. We thought maybe they just appeared kind of out of nowhere. They suddenly evolved this new kind of toxin. But here's some more interesting things. This is all well and good, but here's something that I found more fascinating is we actually looked at these toxins and we're just trying to figure out where they are in terms of like life stages or the tissues. So all that stuff I was talking about with Hydractinia, can we see where jellyfish toxins are expressed in an animal? And can that tell us something about what they're doing for these not box jellyfish? So when we looked at this group, so these are 3 different kind of jellyfish stages for 3 different species, including the moon jellyfish here. We looked and found that consistently when, if we looked at essentially a jellyfish versus a polyp, that I showed in the beginning, JFTs were highly expressed, were higher in jellyfish. So that's a little bit weird. That tells us that jellyfish toxins, these really potent toxins, seem to be beneficial when you're actually moving around in the environment as a jellyfish, and maybe not as important when you're just a polyp stuck on the floor, the seafloor or whatnot. We also looked at the very specific kinds of stinging cells in hydra. So this is the freshwater hydra that are used for prey capture. That's those really scary harpoon-looking stinging cells. And it turns out JFTs are higher in those than the other kinds of stinging cells. So jellyfish toxins seem to be important for catching prey, at least in this one species. And then the real kicker of this whole thing, my animals, Hydractinia, have several jellyfish toxins. So not only is that somewhat frightening, but they are higher in the prey capture polyps. So these feeding polyps and then that predatory only polyp, we found multiple JFTs that are super high in those two polyps. So jellyfish toxins in box jellyfish may be really important for catching fish, but overall they might be really important for all of these species to capture food. They're just not super potent, at least towards humans, because they're not catching fish. They're catching other kinds of zooplankton or other inverts and whatnot. So this is super, super cool and something that I'm hoping to continue to work on. And I just wanted to show this here. Yeah, across this kind of group that I showed before, there are 6 different jellyfish toxins. That's getting on par with box jellyfish for the number of these toxins that are actually being expressed in an animal. And some of them are pretty closely related to those dangerous box jellyfish ones too. So that's something we're working on right now, just to kind of see maybe how potent these toxins are. That's some of the work that I'm hoping to finish out, be the last stint in my PhD work. Okay, I know I'm hitting towards the end, so I will sit for questions, and this is the fastest one. So, all right. So this work is actually something that was not part of my PhD. This is on stinging mucus. I don't know if anyone has heard about stinging mucus, but this is work that I did at actually Washington, D.C. on these jellies called upside down jellies. So they quite literally, as a jellyfish, they don't swim around. They hang out upside down for the most part with their tentacles facing up. So the reasoning for that is that they actually, like corals, live with symbiotic algae. So these almost like plant-like animals. And those algae can get energy from the sun through photosynthesis, and they transfer some of that energy to the jellyfish. So there's actually nice symbiosis where the jellyfish gets a little bit of its nutrients and whatnot from the sun and then catches a little food in return. But they also release these clouds of mucus up into the water that when they make contact with a person who might be just snorkeling or whatnot, looking at these animals, which you can find really all over, it actually causes a sensation of a sting. You might get pain, burning, a rash forming, itchiness, but the jellyfish has never touched you. So how is this happening? That was essentially the question I was asking with my 2 colleagues. And just to emphasize like how much goop you might get. So these are a bunch of jellies that I've put into, I think, a 4-cup Pyrex dish. They have reduced this whole set of water to mucus at this point within 5 to 10 minutes. So they release quite a bit of this mucus out into the water that you might come into contact with. So the first thing that we want to look at, well, what kinds of stinging cells are in the mucus? Could it be that they're just shedding a bunch of stinging cells into the mucus and that's what's firing in there? So they have 3 different kinds of stinging cells, the 2 really small ones, and then these large ones in here that are this purple in color. And you can kind of see here, so as these are just proportions of all these different types, colors as you go from the polyp to a new jellyfish forming to the jellyfish, and then the mucus. The mucus and the actual jellyfish don't actually have that much of a difference, maybe a little bit less of this one kind of type, but not a huge difference. So that was a possibility that maybe they're just shedding a bunch of stinging cells. But when you look at the mucus, there was actually this super weird white speckly stuff that they release in the hundreds sometimes, coming out of the mucus. They were mobile, so we could watch them kind of move around in the mucus or in a dish when we put them in there. And they didn't look like anything. So the mucus traps a lot of stuff. They looked really, really different, very bumpy, very strange looking things. And when you zoom in, they're actually like little balls of stinging cells. So each of these little bumpy bits, these are just like thousands of stinging cells that are coming into these little knobby, weird things. And they were very reminiscent of actually these other structures that we see in that starlet or sea anemone or digging sea anemone that I showed earlier. They have these little stinging cell balls that are called nematosomes. So Cassiopea is the upside down jellyfish, Cassiopea nematosomes. We call these casiosomes. So these weird little stinging structures, and no one had ever described these before. No one had looked in the mucus to see. So we thought, oh, okay, are casiosomes the reason for the stinging mucus? And at first we weren't totally sure, because when you look at these weird structures, they're only made of a super small kind of stinging cell, one that we don't really associate with having a really strong ability to like puncture through a prey item. So we thought maybe not, but they definitely are in large clusters. So there's a potential for them being in such a dense packet, maybe then they can sting something. We were totally wrong to think that these might not be the case. Casiosomes are powerful things. So this video you're going to see this is real time. This is these casiosome structures, these stinging cell structures moving on their own. And then these are brine shrimps, so sea monkeys that we're just going to give just to casiosomes. Remember, these are not animals at this point. These are just like structures that are made by the animal, and they will absolutely sting and kill prey items like these brine shrimp extremely quickly. So they pack a lot of punch into a little package. And so you can imagine that when hundreds of these are being released from one animal, and then you might have tens to hundreds of animals releasing the mucus all at the same time, very likely that that is what is causing this stinging water sensation that has been known for many decades, from aquarists to scuba divers. It might be because of these structures. And just to point out too, we actually looked for jellyfish toxins in upside down jellyfish, and they are absolutely in there, and they are in casiosomes specifically. So we found actually either 2 to 3 jellyfish toxins in these animals too. So definitely have the chemical potential to sting people, and then they have the structures to boot. So this is the paper that came out from it. So if you're into reading scientific papers, this one has a lot of cool images, and it was quite a high number of authors. We got a lot of input from this particular one, but I'm thinking you might also be interested in some of the news coverage that came out of these stinging grenades, little snot-filled grenades, stinging snot, all this fun stuff that came up about these. I'll put a link into the chat too, where I on my website, where I have all the different coverage from these little ones. Was definitely a big thing for a little while. It was very fun to be a part of that too. So again, just the stuff that I've talked about today, just talking about how jellyfish stings, jellyfish stingers, and how they're stinging things, and how we learn more about these. It turns out that, at least in this group, this cool thing called a division of labor in Hydractinia does impact how venom is being used. They do use their venoms for different ecological purposes, like feeding, feeding only, or feeding and digestion. And they are kind of structuring that in a really complicated way. Box jellyfish seem to be really bad because they've had this really distinct kind of trajectory in evolution, probably because they eat fish. It's not because they just adapted to having these novel toxins. In fact, these really dangerous toxins are all over. They're just not super dangerous for people. And then, in fact, yes, jellyfish can go beyond just this contact with the tentacles and the stinging cells, and develop this really weird strategy where they release stinging cells into mucus in order to sting things, which can include people, even though those are not a natural prey item, of course. So I've had been lucky to have quite a lot of support. For those of you that have either know someone in grad school or is doing grad school, it definitely takes a village to raise a grad student. So I've had a lot of support, both in terms of funding and just academically over time. So I just want to recognize those people. And of course, the animals that I've been using all the way. Can't do this without the animals as well. All right, it's a little bit late, but I would be happy to sit and answer any questions. And thank you so much for tuning in. Really appreciate it.
Thanks, Anna. That was really awesome. It was everything I had hoped it would be. I learned quite a bit myself. I do have a few questions in the chat. One of the guys had left, but I think we're still going to ask it. Where do Portuguese man of war fit into the jellyfish phylogeny?
Yeah, so those are siphonophores. So those are in that hydrozoan Hydromedusae. They're actually pretty close to Hydractinia in my animals. So they also have a division of labor. But they're the only siphonophores that are not really deep sea. They're the only ones that live on the surface, right? So they're an odd siphonophore at that. So, but that's the group that they're in. Mm-hmm.
Yeah, I'm sure we often get them on our beaches in the summertime as well. So
yeah, I was gonna say that's probably, I've gotten actually a lot of family that's in South Carolina will send pictures to me. It's like, what is this? I'm like, please don't touch that.
Yeah.
Please don't touch those, everybody.
Yeah, absolutely. So the next one is are there myths versus actual recommendations when stung? Do you know of any actual remedies to being stung?
Sorry, say that last part one more time. Just think about.
You know, there's a lot of myths revolving what to do when stung, like peeing on your
Oh,
skin.
yeah. Yeah, okay. That's what
are actual recommendations?
Yeah, so the most common one I'm sure everyone hears is to pee on it. Don't do that. So that if anything, that will it will either do nothing or potentially make it worse. So let me just start out by using that as an example to say why. So when a jellyfish stings you, so jellyfish have some control over their stinging cells in some way. So they have some control of whether they're going to actually sting something or not. But if a jellyfish is dead on the beach or you just hit it hard enough, they don't have much control over that. And very likely you rip the animal in bits too. So the situation is now that you have a bunch of stinging cells that have fired into your skin. So that's one problem. The other problem is you probably still have stinging cells that are intact or bits of tentacle still on your skin. Most of them are not going to have like fired yet, very likely. So the first thing you need to do is get rid of all of those if you can, as much as possible. So that's a little bit, I think, where peeing on it maybe was thought of as like, it's not going to fire them and it washes them away. It will probably actually do a similar thing to if you put fresh water on it. So fresh water will make stinging cells fire. That's actually how I fire stinging cells in the lab, is putting them into fresh water and just shaking them up a bit. So both kind of the force of fresh water or someone peeing on you and then the fresh water, like the osmotic change, will make maybe more stinging cells fire. So that's bad. You don't want to use really salt water either. That used to be something people recommended, is kind of going back and using salt water because that won't disrupt them as much, but you could still mechanically fire them. For most species, vinegar is the recommendation. This does vary between species. So I usually tell people to look at the local safety website just to figure out what species are there and what's recommended. In the US, though, I think for most species, vinegar, washing with vinegar for at least 20, maybe 30 seconds will both wash away anything that's intact and prevent any more discharge for the most part. And then the second part is what to do with, so once you've been stung, you've been like the venom is in there at some point. So your best bet is to try and kind of stop as much of the damage as soon as possible. And the way you do that is with a hot compress. So a lot of heat, a hot shower for like 20 minutes, essentially as hot as you can stand for at least a few minutes, if not 20 plus minutes. And that will just help essentially your body fight off a lot of those toxins by making because toxins incredibly are not very stable. They will fall apart with enough heat. So
Cool.
as soon as you can. So yeah, so vinegar for most species to wash and get the initial stuff off, and then a hot compress. And then whatever pain, like Advil or whatnot, if you want to take. If you immediately have issues breathing, you might be having an allergic reaction, which is common for folks that also have it for bees and whatnot. That's when you probably want to either get a lifeguard or think about going to the ER. And that would be pretty immediate.
Cool. Well, thank you. What I have a couple more questions, some fun ones. Jellyfish or jellies? What's
I clearly was saying jellies a lot. I'm not super strict on either. So I use whatever whatever gets people to know what I'm talking about. I tend to say jellies just out of habit now, I suppose, but that's very similar to I say sea stars and not starfish. I was just trained to do that as an undergrad when I worked on those. I'm happy with either, as long as you're getting your point across.
Yes, I also was told as an undergrad too, they are not fish, so we should not say that. Funny stuff. Are there freshwater jellyfish? And if so, how abundant are they?
Absolutely are. We actually have some in the lab. I didn't put them in here. So the transition to freshwater is a pretty tricky one. So it's only happened a few times in cnidarians overall. That includes hydra, which is some people, it's pretty common. It doesn't make a jellyfish though, so not really fulfilling what you're asking. There are truly freshwater jellyfish though. They're called, excuse me, Craspedicusta. But if you search freshwater jellyfish, that's probably the first thing that's going to come up. So how abundant they are is actually a tricky question. So not very many people study them. Again, we have them in the lab, and we're one of very few that studies these to any degree. They all Craspedicusta, so all the species within that group, I believe are native to the Yangtze River Basin in China. But this particular species, Craspedicusta sowerby, has, for some reason, that particular one has actually invaded essentially all through Europe. Now at this point, I believe all the continental US states at some point have reported some Craspedicusta at some point, up into Canada, and across several other places. And the reasoning for that is actually when they are polyps, they can make a different kind of stage. So there's an alternate state thing that they can make where a polyp essentially makes a polyp. And they make these tiny little almost seed-like tic-tac things called frustrals. They quite literally are like jellyfish seeds in that you can dry them out completely. And then they go into like tardigrades, kind of like a dormant state. And they can be dried out for days, weeks, years. You rehydrate them and you're going to have a polyp again. And that polyp will be well enough to make more and more polyps. So the thought is that they came either like in water or just in the soils, even of like plants that are used in artificial ponds, of fish that are being transferred over. Perhaps there's even an idea that they came on like the feet of birds that were migrating between. But they're kind of everywhere now where you would find a freshwater quarry. I know there's some like the Walden Pond. A colleague of mine has studied them in there. But we actually found, very honestly, freshwater jellyfish in an artificial pond about 10 minutes from campus, from Lawrence, Kansas. We have no idea how they got there. They were in a full bloom. They were very happy in this really nasty water, and we collected some, and then we have some also from Japan where they actually have them in an aquarium. So you can find them in many freshwater lakes, ponds, and things kind of all over. They do not bloom, or jellyfish don't appear consistently. So you could have a place go 10 years and you know that the polyps are there, or you know there's been a bloom in the past and that they just don't appear for whatever reason. And that's something we're studying, is what are the cues to make them do that, essentially. But yeah, definitely worth a Google, freshwater jellyfish.
Cool. Second to last question, I'll say. So somebody had an interaction with moon jellies, and they were wondering how dangerous moon jellies can be.
So moon jellyfish should almost never be truly dangerous. I know some people, either with sensitive skin or a bit of an allergy, or that are like embed their hand in a tank for a long time. So moon jellyfish can make mucus too. It's not as much or as like viscous and like truly mucousy as the upside down jellyfish, but they can make a tank like pretty like full of stinging cells and pretty irritating, even more so than not. So I know a lot of aquarists over time have trouble with moon jellyfish, but very typically they should not be a problem at all. And that's anywhere around the globe. So moon jellyfish, there's many different kinds of moon jellyfish. I've only heard of a few cases where they've even caused like a rash or something that you would need to apply an ointment or something or take pain meds for. And I believe those are folks that worked in aquariums, so they were exposed over like long periods of time. They were getting constantly exposed and whatnot. So they should not be. So I handle art, and probably shouldn't, but I handle like most of our moon jellyfish with my bare hands if I need to and whatnot. Some people wear gloves, and then that's more than enough coverage for most jellyfish, in fact. So they should not be that dangerous. But I'm never going to say like don't touch a jellyfish. Like when in doubt, just don't touch a jellyfish.
That's a great rule of thumb for most animals.
Yes.
I have one final question. Obviously, a lot of your research is brand new. You're discovering new toxins, and that's just, I assume, the beginning of this research project. Do you see any like biomedical properties to studying all these different types of toxins in jellies, or anything of that nature that may be, significant in the future, like medically significant?
Yeah, so I can talk about two brief examples for some other research that have been done, and then how mine kind of applies. So for sea anemones, there actually is a neurotoxin derived from a sea anemone that is currently, I believe, in phase 2, if not phase 3, clinical trials for autoimmune disease. So that's taken quite a bit of time. That's been like in the pipeline for over as many years as it takes a drug to do. So neurotoxins, particularly in sea anemones, are a major target for new drug discovery. For some reason, sea anemones and corals seem to have more neurotoxins as a larger portion of their venom than medusozoans, these jellyfish-bearing lineages do. So they're not commonly explored for drug discovery in that way. That said, actually the Australian box jellyfish, those same really dangerous jellyfish toxins, there is someone who has actually kind of broken down the exact mechanism for how those jellyfish toxins work. And there's kind of two main things that the ones that attack your heart specifically do. So one portion of them essentially stops your heart from beating. It puts it in a state of, I can't think, it's not paralysis. It's not moving anymore. And then the second part is the part that actually causes degeneration and makes your heart unable to start again. And what they've done is taken that portion that actually just stops your heart, and they're testing it as a way to do more effective heart transplants. So can you use kind of this part of the toxin to put the heart into essentially just a viable but non-beating state, and keep it for longer than if you used than just kind of kept it on ice and preserved. And I don't know where that research is right now, but that is one kind of interesting way to think about it. The way I think about mine is that could actually fall into either of those two boats. I'm not medically inclined, but I want to make it easier for people that are interested in drug discovery or all these different medical applications to know where to look. Right now, for people that are interested in cnidarians overall, just kind of look to sea anemones at random to see what's there. Only now that we're kind of getting a feel for which venoms do different things. Once you start getting into that approach, and then you can kind of use the ecology of the animal to narrow down what might be good candidates for future study. or where you specifically want to look for these kinds of new tools and whatnot. So I think of mine as almost this first step to kind of narrow down how people that are interested in drug discovery look for things much better. People do that already with like marine bacteria and whatnot. And corals a little bit. They can narrow it down by the ecology to see what's a good candidate. But yeah.
Yeah, that's great. Yeah, the reason I ask is I'm very familiar with snake venom being used in a variety of medical procedures, from, you know, fighting breast cancer to diabetes and what have you. And I think that for animals like snakes and jellyfish, where there can be oftentimes bad connotation, that having something like that shows you know, a medical benefit to people can oftentimes change perspectives, something that's more concrete to them rather than just saying, you know, most of these guys are harmless, if that makes sense.
No, absolutely. So I usually point for those things. So like this red fluorescence here. So the original like fluorophore. So the original thing from this was green fluorescent protein. So that like changed the medical field in some ways, because now you actually had a fluorescent thing that you could target in different ways to study these really important medical pathways. And that's from jellyfish. So it's not about venom per se, but I try and say like jellyfish are useful in their own way.
They are. Well, I really appreciate it. I learned a lot. I'm sure everyone here learned a lot. I will have this published on our website, so anybody can check it out at any time.